Well, it is a special pleasure to be here in Copenhagen for me and for my son, because this is our home away from home, so to speak. And, um, and I am really grateful to my beloved former students and colleagues for continuing to invite me to come. So I think my job today is to talk about the ABCD study and a little bit about the HBCD study, though we have a better expert up here to talk about that also. Okay, so the ABCD study can be described just as we put it on our website. So, and our website has a lot of information about the ABCD study. It is the Adolescent Brain Cognitive Development Study. It's the largest long-term study of brain development and child health in the United States. And the ABCD Research Consortium consists of a coordinating center, a data analysis, informatics, and resources center, and 23 research sites across the country. We have enrolled 11,880 children, all between the ages of 9 and 10, and their caregivers, of course. And we are continuing to track them. We will track their biological and behavioral development through adolescence into young adulthood. That's what it says on the website, and that's pretty much what we are. But in the next few minutes, I'm going to try to build a case for this new paradigm, in part by telling you a little more about this large-scale study and very high-dimensional study, and start with the rationale for it. So why do it? Well, first, we've known for decades now, based on neuroimaging studies, that the human brain continues to develop biologically and functionally throughout adolescence, all through adolescence and into young adulthood. And we also know that adolescence really occupies a pivotal position in the health and mental health trajectory of the lifespan. Under the best circumstances, of course, it represents a time of rapidly increasing knowledge and skills and independence and resilience and engagement in creative pursuits. But it's also a period of increased risk-taking, and this can sometimes lead to serious accidents or growing dependence on substances or other sort of damaging behaviors. And it's also the time when the symptoms of some mental disorders first appear. And yet, surprisingly, adolescence is really hitherto unstudied part of the lifespan. But another important emphasis for this large and high-dimensional study is an alarmingly high rate of replication failure in our literature. So what happened? So I'm going to take just a minute to talk about this issue so that people kind of understand what we mean by that. So this is a slide that is borrowed from my colleague. What's that mean? My colleague, who is the biostatistics director of the ABCD study, Wes Thompson. And he defines replicability as effects that are similar if estimated in comparable independent samples. But this graph illustrates some examples of estimates of an association, actually I think I can even make this better here, between an MRI study and a neurobehavioral measure. And it shows the expected distributions of observed rho when the true correlation is a small value of 0.1 correlation. As a function of sample size, let's see, can I do this? There, as a sample size here, which the sample size is between 10 and 100. And because of the large sampling error that you see there in small studies, most will fail to detect an effect this small. But when the same variables are measured sometimes, in small studies all over the world, then a few will produce some of these outlying values that you see in these very wide sampling distributions for small studies. And they will appear to be significant. They will meet conventional criteria for significance, and they will be published. And of course, these estimates, almost by definition, are highly inflated values of the true correlation. And few subsequent failures to replicate these studies will be published. And the result of this is that the literature, our literature, in this case the neuroimaging literature, is heavily biased towards inflated effect sizes. Now, I hasten to add that this problem is not by any means confined to the neuroimaging literature. It is well known to exist throughout the biomedical research literature and has been acknowledged by most people in the behavioral sciences. So this is not just a neuroimaging problem, it's a very broad problem. But note the dramatic difference in the sampling distributions for studies with thousands of observations. ABCD size studies have the power to detect even a small true effect, and the confidence intervals around these parameters are much, much smaller. Now, this is good news for the validity of our results, because not only will effects such as this very simple, small, simple correlation be detected and much more accurately estimated, so will the many parameters estimated in the much more complex and dynamic models that we will inevitably need to account for human developmental outcomes. So in part for these reasons, there's a growing consensus in the scientific community and among funding agencies that large-scale, high-dimensional studies are needed to characterize the ways that multiple factors influence developmental outcomes over the lifespan and to yield more definitive estimates of their relative and combined importance in determining those outcomes. And that statement is kind of a theme for what is emerging from ABCD as we go along. And I certainly won't be able to tell you all about it, but you'll see that this is what is beginning to come out of this study and others of the same caliber, and that is that there are many outcomes that influence, there are many factors that influence the outcomes that we're most interested in. Some with quite small effects, but cumulatively adding up to a considerable amount of the variability. So, just to cut to the chase. Okay, so what are the ABCD study aims? Well, first of all, there was the expectation that we could develop some national standards for normal brain development in this adolescent period, which had not been very well studied. We are measuring individual developmental trajectories, and I emphasize trajectories. We're talking about longitudinal today, so it's very important that we focus on these trajectories of brain, cognitive, and emotional development, and things like academic progress and other things, and identify the factors that seem to influence them for good or for ill, basically. We're looking at the roles of genetic versus environmental factors and of the inevitable interplay that develops between these genetic and environmental factors. We hope to particularly study the effects of health, physical activity, and sleep, and things like that on developing youth. And we will be observing the onset and progression of mental disorders. With this larger cohort, we will certainly see the onset of serious mental disorders. We'll be looking at the variability in the course or severity of these disorders and the relationship between mental disorders and substance use. And back to where the idea for the study really originated, and very importantly, we will be looking at how substance use patterns affect developmental outcomes and vice versa, how different developmental outcomes influence the risk for substance use disorders. Okay, so this study originated, just for a little historical perspective, in an entity at the NIH referred to as CRAN, or the Collaborative Research on Addiction entity, and it involves the National Institute on Drug Abuse, the National Institute on Alcohol and Alcohol Abuse, and the National Cancer Institute, which is for the smoking angle. But as the planning went forward, a number of other institutes became interested in the questions that they could address with this kind of a study, if they added their favorite topics and so on. And so now there are 10 institutes and centers at the National Institutes of Health, and 4 additional federal partners, the NSF, the CDC, the Centers for Disease Control, the National Institute of Justice, and the National Endowment for the Arts. And I think there's another one now, which I've forgotten to add here. But. So just a couple things about the final structure and organization of ABCD. This graphic shows that there are 21 research sites at leading research institutions in the U.S. I know that's hard to read, but we recognize a lot of those institutions. And 2 centers, both the centers are at UC San Diego, the Coordinating Center and the Data Center. And these are funded through 23 cooperative agreements with NIH, which is a little different than most grants in the U.S. And consistent with the provisions of this mechanism, we work very closely with a large number of federal collaborators, at the NIH particularly, but also at some of the other entities as well, from the Centers for Disease Control in particular, and NEA. And though to most researchers this sounds like a bit of a nightmare, you mean our sponsors are actually going to be meddling and working, oh God. But actually it's been really, really helpful. They are with us all the time. They're working on our work groups with us. They're aware of everything that's going on. And they really, the way we like to put it, we brought them our A-team and they gave us their A-team. And so we just have a tremendous group of people working together from the institutes and from the consortium, of course, so that when problems emerge, and inevitably in a big complicated thing like this they do, everyone's aware early on, and we work together to address those. If there are resources that we need, or if there's extra help or extra expertise, they help us to get that. So it's really actually a very nice model. I think we should do more of it that way. Okay, so you can see there are about 190 investigators in ABCD. There are 42 federal collaborators, which tells you about how many people that they have working with us. The research staff is over 500. It's hard to keep track because people are coming in, students and additional research assistants and so on. And we have 23 external advisors. So, large number of people involved. Oops. See if I can get it to go this way. Oh, okay. And that just shows you the national scope of the study, where it is in the, oops, that's not what's there. What's that? What happened there? There it is. The national scope of the study, where our sites and centers are. So, the next thing I want to talk a little bit about is the ways that such a large, highly collaborative paradigm allows us to do things that we could never have done before, including these elements that I'm emphasizing here. This aggressive open science policy that we've adopted and so on. So first of all, it's allowed us to accomplish one of our most important goals, which is to enroll a diverse cohort that resembles, as closely as possible, the entire U.S. population of 9 and 10-year-old children. We were guided in our recruitment by an expert group of epidemiologists at the University of Michigan who provided demographic targets for each of our sites and for the study as a whole. We adopted a primarily school-based approach because all children in the U.S. are supposed to be in some kind of education. And we reached out to the schools specified to us by the epidemiologists, they knew what their enrollments were, and asked them to allow us to provide information describing the study and contact information for the families to reach us. And many times they allowed us even to set up an information kiosk at school events and to come and talk to families there, and that really helped a lot too. But that is how we recruited them. And when we were having difficulty meeting our demographic targets in our catchment area, the epidemiologists would provide us a new slate of schools that was more heavily biased towards enrollments of those individuals who we were not reaching. And so that increased our ability to find more kids who had the right demographic characteristics so that we could really match our country better. So, as I mentioned, there are 11,800 kids who were enrolled. And we did a pretty good job, we think. We have 2 subcomponents of our cohort. This one we're calling the general population enrollment. These are all the school-based enrollment kids. And you can see here this dial that sort of shows you what our targets were on the outside rim and what we actually achieved on the inside. And we did a pretty good job of matching in these very general demographic terms, just as a way to get an overview of this for our over 10,000 kids from the general population sample. there is a mismatch there, but only in the other category, and there's an interesting story there, but I won't tell you unless you ask me later. So. We have another cohort, which is 1,730 kids who were actually recruited as twins, as monozygotic and dizygotic twins, at 4 sites where investigators were very experienced twin researchers, and they're recruited differently through birth registries and twin registries in those states, and they're in Midwestern states, and so our demographic match was more challenging there because they're pretty much Midwestern states, so let's just say that. But we still did pretty well there as well. So these are just the kinds of assessments that we did. I'm just going to give you the overview of the assessment categories at the baseline protocol. All of the protocols that we've administered since the beginning of the study are all described on the website, so you can go and see them all. But this just shows you the main domains. This is from the youth directly. This is the youth protocol. This is our things we're taking directly from the youth. The domains are physical health, mental health, neurocognition, culture and environment. Substance use, obviously there's not much substance use at the 9 and 10-year-old range, but just getting ready for how we're going to assess that. That gets much more serious later. We also collect some biospecimens, some of which are analyzed immediately, and we have the results right away, and others are banked for future analysis. Of course, we have brain imaging protocol, which is T1, T2, and diffusion-weighted imaging protocols, and then for functional MRI, we have resting state MRI and 3 task fMRI paradigms, the monetary incentive delay task, the stop signal task, and the emotional in-back task. There are a few other data sources too. We do actigraphy with the Fitbit. We do geocoding from residential history for lots and lots and lots of linked external data, sort of imputed variables based on the geolocation of the kids, and public databases about pollution, crime statistics, policies, you name it, all kinds of things. We get some information from the teachers, etc. And this is just from the parents. Obviously, in the early years, we had to get a lot of information from the parents, and so this is the same kind of domains, physical health, mental health, substance use, and cultural and environmental factors. The basic visit schedule of the ABCD study is that we do very comprehensive assessments across all the domains at baseline and every 2 years thereafter. These comprehensive visits included this multimodal imaging protocol. And we do briefer assessments at an odd year face-to-face visit. So that one does have some direct assessments, and they're somewhat different. They're not always the same as the comprehensive ones. Again, they're all on the website. And then we do more frequent phone and web assessments. Let's talk about our open science model. It's a really important thing at ABCD. So we actually share, for those of you who are interested in neuroimaging, I guess a lot of you will be, there's just the almost immediate sharing of the unprocessed raw imaging data for authorized users, of course. But more importantly, we have been providing annual curated data releases. So annually, as you can see, starting in 2018, we sort of give a cumulative, we share all of our data cumulatively. So if we've made any changes, found errors, fixed them, changed the processing stream or whatever, we do a new cumulative data release of all of the data, including the interim baseline, the follow-up data, annually, except that on the recent 5.0, we got a bit behind. We're making a transition in our sharing platform, and so we just released the 5.0 in 2023, recently, a month or so ago. But it is there, and this is all on the NIMH Data Archive, sort of data sharing platform at the moment. Okay. So let me tell you about how this works in the ABCD study, the access part. This is what's called a restricted access, de-identified data set, which is acceptable to researchers all over the world who have a U.S. federal-wide assurance. That's just about every institution you can imagine. And prospective users apply online at the NIMH Data Archive, INDA, where they describe their planned uses of the data, and then they sign a data use agreement that prohibits any resharing, any re-identification attempts of any type, and any inappropriate or disrespectful reporting of disaggregated data that would be disrespectful of our communities from which we draw our cohort. And they also agree to report published data, any publications that they do, back to INDA. They then apply for endorsement through their institutions. And this means that their data use agreements have to be co-signed by an institutional signing officer at the recipient institution, which kind of means that the institution has some skin in the game here. If they're standing by this investigator's compliance with the data use agreement. And these are valid for 1 year. And then when you go to renew your access, what we call the DUC, data use certification, the renewals are contingent upon compliance with the data use agreement. So you have to have done all those things that you said you were going to do at that last time. So that's how the access works. So what has the community's response been to this? Well, these data are out of date. I'm sorry to say, we just haven't done a data dump recently, but at the end of 2021, so I know it's a long time ago, but we already had 3,700 unique users who were authorized users of the ABCD data, and they were in 32 countries, or institutions in 32 countries. These numbers are all considerably higher now, we just haven't updated them. Users downloaded an average of 115 terabytes of data per month. And at that time, 181 authors from outside the consortium, compared to 130 from inside the consortium, had published the ABCD data. There are now over 600 publications listed on our website from the ABCD data. We try to capture all of them. We probably don't capture all of them, but we try to. They're supposed to be, we're supposed to be cited and everything. And in 2021 alone, over 142 publications resulted from the ABCD data, more than half from outside investigators. And the other thing is that the institutes have been offering grants for people to just analyze the ABCD data. So you can get a grant to analyze the ABCD data or to combine it with UK Biobank data or other data sets to put it together in some way, for secondary data analysis purposes. And there are many of these grants now. Again, this is out of date now because this only shows 2021. This is something we borrowed from one of our grant officers, but they're funding, they've probably funded about 150 grants. There are probably about 150 grants out there just to analyze ABCD data by other people, and that doesn't count the cooperative agreements that we have to perform the study. So that's just kind of how this particular version of the open science model is working, and I think it's important for people to know kind of how it works and so on. Okay, so you might ask, how do junior investigators fare in this new research landscape? A lot of people were concerned about this early on, within and outside the consortium. And I think it's really important to say that we still and will always need many different types of research paradigms. We all know this, to succeed in advancing the science in our field. However, there's quite a lot of evidence that the junior investigators seem to be doing pretty well working within this paradigm, if that's the kind of research they want to do. And so, for example, graduate students, postdocs, and young investigators are lead authors on a large proportion of ABCD publications. And this is true both for the publications by outside investigator groups and by the inside ABCD investigator groups. And co-mentoring of trainees by faculty investigators at different sites is very common, both formally and informally. And so if someone wants to do something that's on the genomics of mental health trajectories or something like that, they're going to be able to find co-mentors across this consortium who are going to be people working at the state of the art in those areas. And that's often happening in the consortium because we are working together all the time and we know each other. And so I think this is also a benefit in some cases to junior investigators to be able to have a kind of a selection across institutions of their mentors. And one thing we're particularly proud of is this very exciting recent offshoot of the study. It was actually a training program that was launched by two of our PIs at Vermont, Hugh Garavan and Alexi Potter, with Micah Johnson, who's a professor in the Department of Mental Health and Law, and let's see, can I still do this? Let's see, is that it? That's Micah right there, Micah Johnson. He's at the University of South Florida, and the program supports early age BIPOC scholars and provides advanced training and assistance in exploiting the ABCD data resource for their own independent research projects. So they come, they are co-mentored by people from throughout the consortium, and then they develop their own projects, and they get help from anybody that can be from the ABCD consortium to do that. The program launched with an initial cohort of 12 scholars, all together, working together with many different ABCD investigators in Burlington, Vermont, 4 grad students, 4 postdocs, and 4 junior faculty. And it was so successful that the training program has now been expanded dramatically under the same leadership, Micah and Hugh and Alexi, to recruit 80 new trainees over the next 5 years, because it's been really terrific program. Okay, now I'm going to take the prerogative of talking to you a little bit about actual results from the ABCD study. And they're my own postdocs' results, and so I wanted to, because I can talk somewhat more intelligently about those than the 600 that I haven't even read. So yeah, just something, a little example. This is a neuroimaging finding. In this case, well, first of all, it's important to note that a battery of brief cognitive tests, most of you may have heard of it, the NIH Cognition Toolbox, is kind of the cornerstone of our neurocognitive assessment battery in the ABCD study. And there are 7 different tests, and each yields a separate score. And then the tests, there are also some composite scores that you compute to estimate what's called fluid intelligence or crystallized intelligence or composite intelligence or whatever. And they include reading and vocabulary, attention, response, conflict, resolution, cognitive set shifting, processing speed, and memory for a story sequence, and working memory. Those are the constructs. And so to explain our use of the imaging data here, I should define cortical regionalization. This is the pattern of relative sizes of different functional regions in the cortex. This is in essence a pattern describing how much cortical real estate is assigned these different functional areas, right? And we know a lot about how cortical regionalization or arealization emerges in the mammalian brain during the embryonic period, and and in the human cortex. And it's been observed for a long time that individuals show a little bit of variability in this. It's, can be measured in terms of what, for example, what proportion of their total cortical surface area is occipital cortex or visual cortex or something like that. That's known to vary a little bit. So this is a sort of regionalization variability in people. And so we're using the cortical surface area information from FreeSurfer to look at this. And really the observation here is that when one uses a multivariate analysis to predict each of the test scores with the pattern of cortical regionalization across the entire cortex, a distinct pattern is observed for each one of these tests. And they're shown here, and I know it's very difficult to see them. But if one of the things we focused on at the beginning was just sort of this measure of crystallized intelligence, I guess, versus this fluid intelligence. And I think you can see that the patterns just for those composites are very distinctive. They really look very different. And what you're seeing here is that the pattern is characterized by areas which are relatively expanded or relatively kind of restricted or reduced in the cortex. And this is just regionalization, just the cortical regionalization. Right? And again, these are the two. Whoops, sorry. This is the two that are most distinct. But actually, each one of the subtests, each one of the subtests shows a distinct pattern. And those, the differences in those patterns are statistically robust with a whole lot of kids, a lot of data, right? And they're different from each other, and they map differently to these different things. So what does this mean? In essence, this means that the cortex of the best readers is shaped slightly differently from the cortex of the best cognitive set shifters, which is a little bit different from the cortex of the best encoder of a story sequence, right? So why do we think that's important? Well, We think these results challenge conventional theories about this latent causal factor, G, that underlies variation in intelligence test scores. And they're more consistent with another model, which is not ours, but is cited here, explaining cognitive development in terms of functional interactions between relatively distinct neural processing systems during brain development. We think the results underscore the importance of determining how learning experiences of children with slightly different neural architectures influence these interactions and what we might do better to support the cognitive development in all children. In other words, children who have slightly different neural architectures. And so that's why we think this is an important finding. I wanted to highlight one other result. Also, Claire Palmer was the lead person working on this. And this one, again, really highlights why this longitudinal approach is very important, even in the adolescent period. So here we're looking at what we call the restriction spectrum imaging. It's a little bit like the naughty imaging that's already been described. It's this multiple compartment diffusion-weighted imaging model that really separates the part of the diffusion signal that is coming from the restricted compartment, which is more intracellular, and the hindered compartment, which is more extracellular, and free water, of course, but that's not much of what we're looking at here. So this is looking at the restricted normalized isotropic signal fraction. As you heard before, these measures are usually computed as signal fractions of the diffusion signal. And so this one is, the thing that's a little bit complicated is that they sum to 1. So when you see an increase in the restricted fraction, it means there's an increase relative to the hindered fraction. So you don't sure absolutely what's changing, both things, one thing or whatever, but it's a shift. It's a shift in the diffusion signal. So anyway, what we're showing here is what we see when we combine the longitudinal data from the first 2 imaging sessions. So the baseline and 2 years later. And so we're subtending between the differences in ages at the baseline and the 2-year follow-up, the 9 to 14-year age range, basically, in these data. And what we're showing are really just where the age effects are. So we're mapping with the heat map where there are increases in the restricted fraction, diffusion fraction, with age. So it's strictly age-dependent change over that age range, right? And I think it's fair to say, and you can see that some of these changes are located in white matter areas. But really, the striking thing is the changes in these very localized areas in the brainstem, in the diencephalon, and in the striatum and the pallidum. And I think that we would not have expected that between 9 and 14 years of age. I don't think anybody has seen this before. And so now we have to explain it. And as the last speaker said, that's tough because we don't really map the imaging biomarkers to even the neurobiological processes that some people do know about going on in the developing brain. Here we know even less from any of the relevant data in basic neurobiologic studies about what's going on in these structures at the microstructural level. And so we have to explain them. But I would call these discoveries because I don't think anybody was aware that these changes were localized and that they're quite strong. Of course, other things are happening during this period, like the onset of puberty. And so it raises questions about what might be some of the mechanisms that are driving this through the onset of gonadal steroid hormones and their association with some of these structures. So anyway, so this is a discovery. And although I don't have any pictures to show you, we just added the next wave. We now have this, the 4-year data. So that's the baseline 2-year and 4 years. So we have a third time point. And we're not ready to describe the results, but we see this a similar distribution. But It looks to us like there are some nonlinearities here. Some of the structures may be showing a later rise. Some are maybe plateauing earlier, maybe even moving into somewhat different areas. And so we've got a little bit of work to do to really describe that. But we know that this is an unfolding story. So in this adolescent time period, age range, we are seeing things we have not been able to see before. And it's a continuing thing. And it's certainly not at ceiling. So there are continuing changes, microstructural changes, and this is a shift toward the intracellular component. So the diffusion signal is increasingly intracellular in these regions. And there are many explanations for that. We've had some conversations about that, and there are many possible. Could be cell density, could be changing cell phenotypes in these regions, some kind of trophic effect, could be glial effects, could be many different things. So we need to dig down under that, but at least, and from my point of view, it's discovery of these late maturational changes in the human brain and their anatomical distribution. And hopefully, eventually, with those trajectories, we'll really know how they vary across these different brain systems. So I'm proud of that, but that's the kind of thing I'm always interested in, as Katrina knows. So. All right, now I want to say just a couple of things about the HBCD study. This is a Healthy Brain and Child Development Study. And it also can be described well by looking at its website. It says that it is, and it's really small in there, but it says that it is the largest long-term study of early brain and child development in the United States. And if you put them together, it is definitely the largest study of child development, child brain in the United States. And their goal, and they've just launched recently, and they are underway, and as you'll hear more from the next speaker, but the goal is to recruit a cohort of 7,500 mothers and infants, dyads, and follow them for, about 10 years. Well, we all know they'll follow them for a long time. But, beginning in the second trimester when possible. And so that's going to be, even much incredibly heroically ambitious study to accomplish, but they're already doing a great job. I really have a peripheral role in that study, more of a kind of lessons learned from ABCD role. But it's very exciting, and they have a wonderful cohort, and they're definitely going to nail it, but it's going to be very challenging. So, okay, and these are their objectives. Again, this is just saying it's very similar. It's structured in many ways very similar to ABCD. A lot of neuroimaging, especially front-loaded because of everything happening so quickly early on in development. And they are doing many different kinds of assessments: brain, cognitive, and emotional development, neurodevelopmental trajectories, again, across the whole time. A lot of focus on substance exposure. Some of the support from this is coming from the initiative at the NIH to address the opioid epidemic, and so there'll be a little bit of an over-enrollment of children who have been exposed in utero to substances, particularly opioids. But it will also be very largely a healthy brain, healthy cohort. Again, distributed throughout the entire country and recruiting a very representative cohort. Okay, and these are their objectives. There are lots of them, and they're on the website, and so I won't go through all of them, but very similar because there are going to be a lot of interest in the genetic factors. Early life exposure to opioids and other substances is a major theme in the study. Looking at whether there are key developmental windows where the impact of adverse environmental exposures of any kind influence later neurodevelopment. Developmental windows during which ameliorating influences are protective against the potential neurodevelopmental insults of adverse exposures, and what is the impact of early parent and caretaker interactions with their children. I'm losing this. Okay, I'm going to conclude now. So the points I want to make are that developmental population neuroscience, this is what we call this stuff, is a new, highly collaborative way to do human developmental and biomedical research. And the advantage is that many potentially important factors that may influence the development of the child and affect important outcomes can be measured in real time. And the result is a very large and high-dimensional data resource for the entire community. The increase in power means that the statistical effects of biological, environmental, and behavioral factors and their interactions on important outcomes can be accurately estimated. Open science is accelerating the progress and increasing the impact of our work. It's doing so by increasing the number, disciplinary range, and diversity of users of the data, of the scientists who come to the data with their questions, many of which none of us ever anticipated. It's ensuring the transparency that we need to resolve inconsistencies in the reported results, because every paper has to cite the data release and exactly, so that another person can go and get exactly the same data and either replicate it or not. And it's lowering barriers for junior investigators to contribute to the literature with major publications, in some cases, if this is the kind of work that they want to do. Okay, thank you for your attention, and I will take questions if we have time.